Hi, today I'll be presenting about the engineering of the secretory pathway in the yeast species Cleveromyces marxianus for improved secretion of proteins. I'll start by introducing the space of synthetic biology, which is how the field of metabolic engineering came to be. DNA or nucleic acids is the universal language across living organisms, containing genetic information that can undergo transcription, process of copying, and translation, which synthesizes proteins for biological processes. These synthetic gene circuits can be introduced most commonly through plasmids and allow the gain of desired functions in living organisms. Our lab focuses on metabolic engineering, which can be summarized as the study of improving the rates of desired reactions or pathways. We are most interested in applying this engineering in K. marxianus, an important industrial yeast chassis. K. marxianus is the fastest known growing eukaryotic species, able to metabolize a variety of carbon-based sugars as well as being thermotolerant, growing up to 52 degrees Celsius. Remarkably, it also has a very high secretion capacity and has a eukaryote capable of post-translation modifications. And so in this project, we will seek to optimize the secretion cassette for increased protein secretion in K. marxianus. Motivations for this is the importance of secreting proteins into the extracellular environment. It allows for proteins to properly fold, mature, as well as the function of synthetic circuits in the environment, like bioremediation. Another crucial application is the ease of harvesting proteins, reducing downstream processes and overall costs in biotechnology. And so engineering the protein secretion pathway is linked to the main processes shown, summarized as the recruitment of proteins by signal recognition particle to the ER lumen, followed by translocation to Golgi apparatus, and finally the extracellular space. We conducted our research by first designing our secretion cassettes with N-gluconase cell A as test chemical and transformed them in our K. marxianus strains. This was followed by growth and protein quantification by an absorbance assay. The parameters we tested were origin of replication, signal peptide leader, strains, and carbon substrates. Finally, we incorporated the novel approach described in our group's previous work with Baker's yeast, where we replaced the CYC1 terminator with PMP1's untranslated 3' UTR sequence, a protein observed for SRP pre-recruitment. Our main consideration for this is to promote co-translational translocation, as the alternative post-translational may be much slower. Our results show that growth and secretion of cell A is generally better in dextrose than lactose and in strain CBS6556 rather than 712. By picking the strongest signals, we also tested for the pre-recruitment of SRP. It showed that PMP1T showed a great increase in the INU1 signal with an increase of about 80% while not affecting OST1. We also observed that plasmid origin affects our strongest signal OST1, where PKD, which has much higher copy number than CENARs, resulted in greater secretion close to a 5-fold increase. This summarizes our findings for optimizing secretion cassettes, and moving forward, we'll try rational strain engineering to further boost protein secretion. Thank you.